So thanks for coming to our presentation. We are interested in combating systemic fungal infections by targeting the Achilles heel of fungi, which is in their transcriptome, in their RNA molecules. Well, okay. If Mary says so, I better do it. So it's been founded by myself and Kevin Polizzi, and we have a wonderful set of business advisors and a terrific scientific advisory board. The overall company view is that our goal is sort of summarized in this picture here. We want to target RNA molecules that are unique to fungal organisms and do that with small molecules. And we are doing that using a patented high-throughput assay that was developed by people in my lab. And in this way, we are specifically targeting RNAs that are found in fungi and are not in mammals. And so we have a new solution for systemic fungal infections. We have broad expertise in RNA targeting and infectious disease, and our validated platform is going to be extensible to human-specific diseases as well. So we heard a little bit about this before, but the need for antifungal solutions is growing rapidly for a lot of reasons, for all of these important indications, but people may not realize a growing threat is the rise in infections that come from things that get implanted into the body. So implants, transplants are frequently complicated by systemic infections, and also immunocompromised individuals are particularly at risk. Fungal infections are really hard to kill, and the reason for that is that fungi are like us. They're eukaryotes. We share many proteins with them, we share a similar cell structure, but our RNAs are very different. But as a result, available antifungal drugs can be really toxic because they're going after those sort of traditional targets. And to meet this need, we've developed a set of compounds called IntronX that target RNA enzymes unique to fungi. So why is this important? It's important because existing drugs are not meeting the needs of patients. Many of them, as you can see, have been around for 50 or 20 years. There are many from the same compound class. They tend to be highly toxic, and even those that are well tolerated have to be taken IV. As a result, we just don't have many solutions in the clinic. There's very high mortality, growing resistance, as we heard about, and emerging untreatable pathogens that we read about in the news and actually now are watching on TV. So our strategy is to create new classes of small molecule antifungals that maximize efficacy and minimize toxicity. So how are we going to do this? We really want to target this unique class of molecules that are only found in fungi. So it turns out that many fungal genes, the mRNAs from those genes, are interrupted by little pieces of RNA. You can see the black line there. They have to be spliced out before the gene is functional. In fungi, that is accomplished by the fact that they form specific 3D structures that can be modulated and will respond to small molecule drugs if they're highly specific. So interestingly, those fungi of concern I'm showing you here on the left all contain one of the same molecular types that can be inhibited by similar molecular scaffolds. So what we do is that as a splicing pathway proceeds and the pieces of RNA are brought together, we have a sensitive molecular beacon that can actually catch it in the act and watch as it's inhibited by small molecules. And it does that by unfolding when splicing occurs and base pairing across the boundary of spliced genes. This is a very sensitive assay. It has many logs of sensitivity, and this enables us to screen huge compound libraries for effective inhibitors and also to do SAR and optimization. So this has already produced novel antifungal compounds. The version 1 compounds have been published, so I won't tell you too much about them. These target a very specific class of fungi. They're very potent, and they have an MIC that's comparable with the clinical standard of care, and they're active in vivo in bloodstream. What we're really excited about is our version 2.0 compounds, and I'm just showing you the inhibition curve for just a hit, something that's not even been optimized yet, so very potent, submicromolar. And then one thing that's really exciting is that in the lab now, we're solving the cryo-EM structures of the fungal targets with these small molecules to really jumpstart our SAR and to help us with optimization. So we're really moving quickly. And so we have started this company. So our business model is that we want to license existing assets that we have. We're going to use our lab to develop new ones and to extend our platform to new kinds of antimicrobial and mammalian targets. So right now what we're trying to do is raise our seed funding, hire our executive team, and transfer the knowledge to our corporate lab. I want to thank you and just close by saying if you're interested in RNA targeting and you want a very well-characterized target and system in which you want to get into that space, I think this is a good play. So thank you very much.